today's topic is systematics. We have studied taxonomy in detail. We have studied hierarchy of taxonomy, classification, method of identification, binomial nomenclature. Now, this term existed ever before taxonomy, and in present scenario, present time, generally it is termed as synonym of taxonomical classification or taxonomy, but in real it is a larger dimension than taxonomy. How? So in 1970, a scientist Meitner make 6 points to differentiate systematics or biosystematics from taxonomical classification or taxonomy. What are those points? A. Identification and naming of a species. What we do under systematics, point A, identification and naming. B. What is the second point? What we do, the second task in systematics is description of a species. Description of species. What is the third task which is done under systematic? C. Classification of the species under taxonomical rank. Proper classification. The fourth task which we do under the heading of systematics is preserve the specimen. Preserve the specimen. E. Study their phylogenetic relationship. Study their phylogenetic relationship. And F, which is the sixth point, consider about the environmental adaptation of that species, and that comes under consider or study their environmental adaptation. So we can see here easily that identification, description, classification and preservation, all these four things we have studied under the heading of taxonomy. But these two things are new to the taxonomy, which will be headed under phylogeny. That means taxonomy plus phylogeny is systematics. So systematics or biosystematics or botanical systematics, whatever the term which we use is the diversification of life form, presentation of diversification of life forms on earth on the basis of pleisomorphous characters and apomorphous characters. So Now I think you are clear that how systematics is different from taxonomy. Systematics is not different but just a wider dimension, uh, which have to add the phylogenetic study in taxonomy, then it becomes biosystematics. Again, what are the branches of systematics? What are the modes of systematic biosystematical studies? So there are three branches, three disciplines in biosystematic, which we will see here, and these are numerical systematics. This is also known as biometry systematics. Biometry systematics. The second one is known as biochemical systematics. And the third one is experimental systematics. This is also known as evolutionary systematics. So we will see it one by one. First of all, numerical systematics. Numerical systematic focuses upon pleisomorphous as well as apomorphous characteristics and keep no distinction among them. So, numerical systematics is based upon biological characteristics, morphological characteristics, and focuses upon the biological characteristics and keep no differentiation among pleisomorphous and apomorphous characteristics. What are these two terms? Pleisomorphous is the term which we uses for the shared ancestral characteristics. Numerical taxonomy is based upon biological characteristics. Biological characteristics has been divided to two parts, pleisomorphous and apomorphous. Shared ancestral characteristics is called pleisomorphous and derived evolutionary characters are called apomorphous. So, numerical systematics compare these traits and find out closeness between a taxa among the group of the taxa or individuals, and on that basis they design the phylogenetic tree. But what happens sometimes, the numerical taxonomy get misled due to convergent evolution. The taxometric unit which numerical taxonomy uses is known as phonetics. Phonetics is that taxometric unit which never make distinction over pleisomorphist and apomorphist characteristics in designing a phylogenetic tree. So such type of work sometimes get misled uh, due to convergent evolution. What is convergent evolution? We have studied it in class 10th that organism from different ancestral stock, organisms from different ancestral stock develops similar trait in similar habitats. So this is the demerit of numerical taxonomy. Now we focus upon biochemical systematics. What is this biochemical systematics? Biochemical systematics focuses upon those units within a cell which make it a living unit. What are those units? Those units are nucleus, nuclear organization, cytoplasm, cell organelles, these are the traits on which biochemical systematic is based upon. Now, the third one is experimental. Experimental systematic focuses upon evolutionary traits. Evolutionary traits such as mutation, gene flow, genetic drift, mutation, gene flow, genetic drift, in designing the diversification of life form on Earth or designing a phylogenetic tree, experimental systematics uses these traits. So on the basis of using traits, the systematics is designed into three forms. Now there are two terms which comes as rival in the field of systematics. These two terms are phenetics and cladistics. Phenetics and cladistics. What is the difference between phenetics and cladistics? Both are taxometric unit. Taxometric unit. This one is 2. Taxometric unit. This is used in numerical taxonomy. This is used in the case of biochemical as well as experimental or phylogenetic taxonomy. In phonetics, there is no differentiation among pleisomorphous and apomorphous characteristics. No differentiation. No differentiation upon pleisomorphous and apomorphous. Here, the differentiation is considered. Differentiation is considered in designing phylogenetic tree. Phonetics has been superseded by cladistics in present time, because we have seen the reason is clear, very much clear, that due to convergent evolution, the phonetical study can be misled. And again, it is also based upon the similarity among biological characteristics only. So we cannot achieve at right conclusion by using only biological traits always. So similar ancestral species can be grouped into different divisions or taxon. That's why this practice is superseded by cladistics taxonometric unit. Now I think you are much clear. We do systematical study through cladogram to differentiate the relationship among organisms, which different group of organisms which have common ancestry can be represented through cladogram. The branching pattern and the differences and the length of the branch is remarkable in designing the cladogram. A cladogram shows whether two groups or two individuals are monophyletic or paraphyletic. So cladogram shows that whether if a line has been shown in such a way, then this shows retrogression. Retrogression means moving towards ancestral characteristics. Now B and C are monophyletic, whereas A and C are not monophyletic. Here D and E. D and E are here monophyletic. But here a retrogression has appeared that this species is moving towards the characteristics which he has gained is similar to the ancestor. then such inclination towards the ancestor is known as retrogression. Okay, I think you all have understood what is systematics or biosystematics or botanical systematics, how is it different from taxonomy, what are the branches or disciplines in which we study it, like numerical taxonomy, like biochemical systematics and experimental systematics. What is the difference between phonetics and cladistic is clear. Phonetics and cladistics both are taxometric units which is used in different disciplines of systematics, whereas we know the fact that phonetical practices is superseded in present time by cladistic. Cladistics uses cladogram to show evolutionary relationship among individuals and group of organisms. In designing cladogram, the remarkable thing is the branching pattern and the length of the clades or branches, which shows that how the groups are have similarity or dissimilarity from one another. So I think this topic is clear to all of you. Go through the topic, note down in your fair notebook, learn also from the book and the side book, and if you have any doubts, contact me and clear that doubt. In next session we will see the new chapter. For this chapter I will provide you assignment. Till then, goodbye, stay at home, stay safe.